Welcome to Lecto. Today we are going to discuss viral growth curve. So let's start. First of all, we will discuss generally that how virus infect the different cell. So if you see here, like this is the virus, okay, and here if you see like this is the cell. First of all, the virus will attach to the cell and penetrates its genetic material into the cell. So if you see here, like this virus will attach to the cell, okay, and after that it will insert its genetic material into the cell, okay. No doubt it will insert only the genetic material, but in some situation you will see the virus can enter the cell as a whole. So after that you will see the genetic material of the virus will take over the control in this cell, and the function will be, or you can say instruction will be given by the genetic material of the viruses. And what actually message comes from the genetic material of the viruses? So in this case the viral genetic material give instruction that it will make that the cell make different proteins for the viruses, okay. After that you will see the formations of the different viruses inside the cell, okay. Now if you see here, like in the cell you will see the lot of different viruses will produce, and after that the cell will rupture, in this case rupture, and the different viruses will produce, okay. If you see, notice that only one virus entered into the cell. After its replication, you will see the lot of different viruses. So approximately, if I say like in the 10 hours, you will see one virus, or you can say virion, produce hundreds of new virion. So this is the general perspective that in the 10 hours you will see one virion will produce hundreds of the different new virions. So this is the general case that how virus infect the cell. So if we discuss the curve, so first of all we will see what actually present on the x-axis and on the y-axis. So if you see, like this is the x-axis, this case is the x-axis, okay. In the x-axis we take the time in the hours, okay, but on the y-axis if you see, like this is the y-axis, on the y-axis we will take the virion our cell, host cell. If I speak specifically, this is actually the host cell to which the virion will attack or infect, okay. This is actually a ratio between the virion, or you can say host cell, or you can say fraction, okay. If I say that there is one virion and there is one host cell, then the ratio will be the, or fraction will be the 1, okay. If I say that there are 100 virion and 100 host cell, the again ratio, or you can say fraction will be the 1. So here is the answer 1. This is the, if I say that the virion, number of the virion is actually again 100, but the host cell, if I say that 1000, so if you see 2 0 cancel each other. If I divide 1 divided by 10, the answer will be 0.1. So this is the second case, and you see all these numbers are actually calculated in this way. Don't get confused, these are not the number of the virus, because it's a fraction, and we cannot say that the 1 virus, or you can say 0.1 virus. It's wrong to say that 0.1 virus. It is actually a fraction. So that's why the number is actually present in different fraction, okay. Now if you see, first of all, if I say that the starting point will be the 1, it can be different ratios, like if the virion, number of the virion and the host cells are equal, then you will see the 1. Let's suppose if I consider there is one virion and one host cell. So the starting point with the passage of the time, okay, now the time will be in this way and the ratio will be the in this way. Okay. When the with the passage of the time, you will see the virion disappear. Okay. If I say like in between the 2 and the 3 hours, you will see the virus disappear. How? Why virus disappear? If you notice that when the virus enter into the cell, okay, now the capsid will remain outside, only the genetic material will enter into the cell. And during when the genetic material enter into the cell, the genetic material take control the whole cell, and in this phase you will see there is no virus, there is no detectable virus inside the cell. So that is actually the intermediate phase in which you will never detect any virus. So that's why with the passage of the time, a state reach when you will never detect any virus. Okay. If I say like after some times, okay, in this case, let's suppose if I say after 5 hours, you will see that here is the case. The virus start detecting inside the cell, and you will see with the passage of the time there is a extreme increment in the virus number. Okay, this is the case after 5 hours. Okay, this is the case. In this case, you will see two periods. The periods in which you will never detect any virus. So, at the starting phase, the period in which you will never detect any virus inside the cell, that period is called eclipse period. Okay, this is the period. You will never detect any virus inside the cell. Okay. After that, you will suddenly see that there is an increment. With the passage of time, there is an increment in the number of the virions. So, this is the second phase, and that's why it is called the rise period, in which you will see the increment in the number of the viruses. So, this is the case. But keep remember that when you will see there is no detections of the virus, but in this period you will see the genetic material is still working in this period, and after that you will see the lot of different virions, and you will see the burst of the cell, and you will see release of the different viruses. So in this case you can also draw another curve before the again replication, or before the rising period. You can see the another curve, and that curve for the genetic material, or you can say nucleic acid, because before the rising period nucleic acid is still working, and still genetic material is working on the manufactures or the productions of the different viruses in the cell. After its production, you will see the detection, or you can say the different productions of the viruses after the nucleic acid. So this is the curve for the genetic material. Okay, this is the genetic material, and this is for the viral. So hope so you got the point that how different phases interchange. Okay, there is a term that is quite confusing, and that term is actually, you will see, latent period. What does it mean? In the latent period, it is actually, you will see the starting of the infection. This is the case, starting of the infection and the state at which you can detect the virus or virion outside the cell. So this is the case, starting of infection and detection of the virus extracellularly. This is the most important point. So you can only detect viruses extracellularly when you will see the burst or the ruptures of the cell. This is actually called the latent period. Okay, but the eclipse period is the phase at which you will never detect any virus inside the cell. So actually I can say that the eclipse phase is actually some lie in the latent period. So this is actually the difference between eclipse period and the latent period. Okay. Next is the most important thing which you have to remember is the cytopathic effect, or you can say CPE. This is actually the state in which you will see the lot of morphological changes in the cell. No doubt morphological, you can also see the functional effect as well. So this is actually the cytopathic effect. So if I say that when the virus enter into the cell and take control inside the cell, and after cell you will see the cell rupture or lysis, you will see the lysis of the cell. When the cell lysis, you can say that lot of changes will happen in the body, and you can easily detect the that part of the body in the light microscope. So this is actually the cytopathic effect. No doubt all viruses create the severe cytopathic effect. So this is the case. Cytopathic is simply is a morphological thing that can be seen by the different light microscope. So this is actually the cytopathic effect that is caused by the different viruses. So this is all about the viral growth curve. If you have any question, you may ask in the comment section. Thank you so much.